The picture you see before you is actually here in Hawaii and was taken by my husband. You're going to see a lot of great photos because he does a lot of great cave photography. So we're going to start with making it go to the next one. Okay. So this is a slide provided by my colleague and friend, Penny Boston, who works for NASA. She and I have worked in caves together for several years, going on like 2.5 decades. And early on, she introduced me to the idea that caves weren't just here on Earth. They're actually all over the solar system and probably elsewhere in the universe. And one of the things that she did way back in 1992 was she wrote an article that proposed that if we're going to find life on Mars, it's probably going to be in the subsurface, and that was a really good place to look. And in fact, there's been a lot of research, a lot of imaging of Mars and the moon and and other bodies in our solar system that have shown the presence of lava caves throughout the solar system. And so that's one of the targets that we are looking at. If anybody wants to know more about the work, she has an article on extraterrestrial caves in the Encyclopedia of Cave and Car Science, as well as lots of other articles throughout various venues. So why we care about caves and looking there for life. Well, one of the big things that she proposed in that article and since is that if life exists in these different environments or remnants of life in our solar system, they're probably going to be in the subsurface, which is much more protected. It's protected from ionizing radiation and storms on the surface and many different factors that could make it really difficult for life to exist. And I don't know about you all, but I really would like to believe that we are not alone in the universe. And in fact, there's lots of different studies that suggest that we are not. So I have actually studied microbes in caves since the 1960s and have branched out to not just the United States, but several different countries that you'll hear about, Germany and the Azores and Mexico and Iceland, looking at different cave environments, and things do differ around the world, which makes it all the more interesting. And the microbes in caves are interesting for a couple reasons. One is there's a whole different life that exists in caves. In fact, one of the things we've shown in our studies is that sometimes as little as 2% of the microbes that are in the cave overlap with what's in the surface soil above the cave. Lots of new capabilities to be discovered and lots of new life, but it also leaves behind remnants of the life that give us clues as to how to find life elsewhere. Caves are also a great geomicrobiology laboratory. So this is looking at the interface of geology and microbiology, and this is how rocks provide nutrients and energy for the microbes, and how the microbes change the actual rock itself, because there's a lot that happens in these interactions. And I'm going to give you some examples of this. And these caves are great analogues for life on other planets or the remnants of life and how we find that. So how are caves different from the surface? So as I mentioned, it's dark. Once you get beyond what's known as the twilight zone, it is completely dark. You cannot see your hand in front of your face if you turned off your light. And it is very quiet and very dark. It is an amazing place. I've done trips in Lechuguilla Cave where we camp for like 4 or 5 days and sleep in the cave. And it's an experience really beyond just anything I've ever had. It's also really human, which for some of the life you're going to see is really important. The temperatures are relatively constant. in New Mexico, ours are, well, in general, cave temperature is the mean annual surface temperature. But in the New Mexico, some of the caves, like the one you see in front of you, they are actually cold traps and are actually a good 10 degrees or several degrees colder than it is on the surface. A lot less to eat. There is no photosynthesis beyond the entrance twilight. All right, so one of the things is that in caves, there are what I would call really obvious microbial things, deposits. So over here on the left, all this pink that you see is large microbial mats. And what those look close up is what you see over here on the right. This is a microbial colony in the Azores on the wall. And all these are water droplets because one of the qualities they have is they are hydrophobic. And so they actually repel water. And so this makes the cave look sort of silvered when you look at a white deposit all over the wall. So our challenge is we need to be able to tell an astronaut or a robot or some other device that's going to sample for us what they need to sample. And one of the things we've discovered over the last 10 years is how much life, microbial life, is actually present in what I would have called secondary mineral deposits in caves. So these are deposits that grow after the lava cave is formed, and they can be things like these little polyps or fingers that you see on this rock. They can be mineral crusts like you see in the background, or several others that I'm going to show you over the next slides. And so our real challenge is to identify what are the best targets for sampling on an extraterrestrial body. So I'm going to take you in various places around the world where we've done some of this research. We're going to start with Hawaii and research that we're doing here. And then we'll also look at some New Mexico and things that we have done in Iceland with Arne Stephenson. And also then in the Azores with my colleague Maria, we call her Lourdes Depkavovich. And you see some really unusual formations here and over here from the Azores. This is work that I've done with a cast of all kinds of students. I'm going to start with some work I'm doing here in Hawaii with Joe Medley and Mike Spildy and Penny Boston. And we'll look at some others. So these secondary minerals can take various forms. Some of you know them as various calcite, calcium carbonate formations you see in lava caves. Those of you who've been in the caves, there are others that are made of gypsum or other minerals that are there. There's a lot of silica in secondary mineral deposits. And what we've been looking at are the different shapes and which ones are most likely to be good places to see the life. So here we're going to look at various linear ones. A lot of them are knobby. There are even ones that look like a head of cauliflower. Some of them look like little pimples on the rock, or coralloids like you might see in the ocean. And the textures can be flat or bumpy. You can have rims. And the really amazing thing is there actually is quite a variation in the colors that we see, purples, golds, yellows, oranges, turquoise. And we're going to go through some examples so you can see the wealth of what's there. So we're going to we've looked at ones in Hawaii, New Mexico, the Azores, even Australia, which has some amazing ones. And we're going to look across these because they have some variety. They occur in basalt. And one of the things that's important for the life that we're going to look at is that they are not that far below the surface. You're talking about a few meters to maybe 10 or 20 or 30 meters below the surface. Many of them are geologically young, although one of the ones I've done in Hawaii was 60,000 years old. But most of the ones I've done, the ones actually near where we live here in Hawaii, are under 100 years old in a few cases, and they have lots of secondary minerals. So this is one of the caves near where I currently am, called Kulakai Cavern. This is actually a commercial cave that you can go and visit. And this is Joe Medley. I'm going to show some of his work. And Joe and I and others together have developed what we call a mineral-microbe continuum. And we're looking along that continuum to see what are the best sources for finding life or its remnants. And so this goes from mineral crust. I'm going to talk a bit about the blue-green ones that you see here in the top, which are copper minerals. There are a lot of iron and manganese crusts that we see in the cave. This is an example of sort of knobs/coralloids that you see on the ends of formations. And these are what we call spheroids. These I have only seen in Hawaii so far. So I'd be interested to know if people have seen them elsewhere. This is a wonderful enigma, and I ask you as I talk to think about this, because this yellow, this what I would call butterscotch, is what we call ooze. And I've been told by people ooze is not a good name. It's already in use as diatomaceous ooze in the earth, bottom of the ocean, on the ocean floor. So I'm looking for ideas. If anyone has a different idea what we should call it, I'll show you some closer images later. And then this is one the Europeans will probably appreciate. This is what's known as moon milk, or and I'm going to mispronounce this, Monmilch is what it used to be called. I believe that's German. Correct me if I'm wrong. But it was used in the Middle Ages in Europe as a wound healer. And the reason is there are a whole lot of bacteria in it, in the group actinobacteria. And these produce antibiotics, and were a way to make it so that the cave or the wound would heal of the infections that were starting. And then the thing that I started studying with Penny and Mike is the microbial mats. And so what you're seeing here are white microbial mats that are also having yellow microbial mats grow on top of them. So this is a continuum that we're going to look at examples of. So this is in the cave we've been working on in this trip. And my thanks to Jim and Lynn for the permission to visit this cave and to take small samples. This is called Maelstrom Cave. It is a section of the Kanahina system, which is tens of kilometers long in the subdivision. And it is the one where we've done a lot of study of these blue-green minerals. And this is an example of what it looks like close up. This is a couple centimeters long, even though it looks much bigger. This one is many centimeters long. These are in two different caves here on the island. And when you look at the names of microbes that we have been able to genetically sequence out of here, what you can see is if you look at the beginning of several of these words, they start with nitro. This is an indication these guys are involved in nitrogen cycling. The ones that start with thio indicate that they are bacteria that utilize sulfur. And then the methyl indicates that there is some involvement either with producing or utilizing methane. So lots of different things they do. We did an article on this. If you care to read more about it in 2011 in Astrobiology, there's an article on the astrobiological implications of some of the cave formations. And so Kenneth's photo actually made the cover of Astrobiology, which was lots of fun. If you look inside these blue-green deposits, this is what you see with a scanning electron microscope that magnifies this hundreds, thousands of times. And these are clusters of microbial cells. And then if you look at the string-like appendages, which, by the way, are less than 1/100th the width of one of your hairs on your head. And these when you look at them close up, are what we call reticulated filaments. And there are some Spanish researchers, Ana Miller and others, who have also looked into these. There is a cave in Germany that we work in, and I'm going to mispronounce it, to Advent Hole and Herbst Labyrinth, where there are just hordes of these little reticulated filaments. Each of these is less than 1 micron wide. And even though it looks like this ought to be a mineral coating on it, this is actually all carbon when you put the electron beam on it. And these are found in those caves in Germany in incoming mud and in the pool fingers that are forming in pools there. Mike Spilde looked at it with a technique called XRD, which allows him to analyze the elemental composition and the minerals that are in it. And it is a poorly crystalline chrysocolla, which is a copper silicate. This is an SEM that Joe did where he looked carefully at the inside of some of this blue-green, which you see over here on the wall, it forming in a crack. And here you see a biofilm. You see a lot of these filaments. And if any of you happen to know what these balls might be, we don't know if those are fungal spores or what. If you look at the micron bar down here, this is a 10 micron. So you can see these are anywhere from probably 4 to almost 10 microns in diameter. And we don't yet know what those are. I'm going to next show you an analysis where we put the electron beam on it and have it analyze the elemental composition. We're going to look at a spot here, and we're going to look at one of these balls. And the intriguing thing is, when you look at that mineral that I first pointed out, these are the elements that are present. And by the way, we coat it with gold and palladium, so you can ignore those two. And there is vanadium, a rare earth element, and there is bismuth. And this is the first time I've ever. So the next thing is these are large spheroids that we see on the walls of Maelstrom Cave. And the interesting thing when we looked at them in the SEM is they're actually gypsum, which are what some of the secondary minerals are. You can see here, this is a 500 micron bar scale. And I seem to have lost my cursor. Can you still hear me okay?
Yes, we can.
Okay, I just have lost my cursor. Okay, well, you can also see over there on the lower right, there are there's a little bit of filamentous microbes there, so those are present. So even though the spheroid looks very mineral, there are microorganisms present in it. So this is the fluffy moon milk, which I mentioned was used for wound healing. And it comes in a fluffy form. And when you look inside of this, there are all these different microorganisms. They're also for the geologists in the audience, if there are some. This is a very, there are some specific mineral forms, like lublinite is one of the minerals that you see in moon milk. And notice all the filamentous microbial forms. And here in the lower right, you can see a lot of the spheroid shapes, lots of different things going on here. And it's just full of microorganisms. This is one of the really cool deposits that we study. And then we're going to turn to the ooze, which again, you might put some comments in the chat if you have other ideas for what we might call it. And you can see a closer view of the ooze right here. This is a butterscotch colored one. There are also ones that are more of a cream colored ones that we see. I first saw this in the Azores, and it was sort of an orange red. And it was just really dramatic. To me, it looks like somebody took a jar of baby food and threw the contents against the wall. And you see this a lot. We also have the reticulated filaments inside this, along with several other microbial shapes. Turns out the ooze, and you'll see this when we get to a little bit more of the composition microbially, is full of all kinds of different life, and it is different than the secondary mineral or the microbial mats. And then finally, the microbial mats. This is an example of some of the white microbial mat, and lots of these spheroidal shapes with lots of little hair-like projections on them that you see a big mass of them over here. So one of the things I'm doing with my German colleague, Andreas Fletch, who is studying all over the world the microclimate of caves, and he's also working with others at another German university, Thomas and Michael, who are studying the geometry of the cave. So how do the dimensions of caves impact the microbial life you see there, and how does the microclimate affect it? So Andreas puts all kinds of very sophisticated instruments to measure temperature, relative humidity, and airflow. And so we look, for instance, at what it's like close to the wall where many of these microbial formations are versus the middle of the passage. And this is a cave map of the passages in Maelstrom Cave. And we see that the red here at the entrance of the cave is very dynamic. It's constantly changing. Whereas when you get down by the wall or even in the middle of the room, there are changes, but it's a much lower amplitude of what you see. And the relative humidity as you get next to the wall is really the yellow that you see here is much more uniform. And so for a lot of microbial life, this is a better place to be. So now we're going to travel on Hawaii up the slopes of Mauna Loa. And we're looking across, by the way, at Mauna Kea here. You can see the if you look really closely, you can see the telescopes on the surface, on the top of Mauna Kea. And we went to lava caves here. And there's something really dramatic you should notice about this one. And that is there's not a green leafy plant anywhere in sight. So the nutrients that reach inside the cave are really different, and it's also much colder up here. There's actually ice in these caves. And microbially, things are really different. Go get me a Pepsi. What you're seeing here are things that look much more mineral in this, and again, you're seeing lots of organisms, even though it's pretty mineral here. A lot of these use nitrogen or sulfur. And this is actually an iron utilizer. This one can actually be an ammonia oxidizer. So lots of different things that these guys can do. And we do have moon milk up here. But if you look really closely, you'll see this is a much drier moon milk. And so different in texture, and looking at the organisms that are present, these are not ones that are known as rock eaters. These are more carbon utilizer for the most part. So we have very different life at different elevations and different climate zones. But there's a really intriguing thing that's present here. If you look really carefully up here at the yellow sort of gold deposit that's here, that turns out to be a vanadium oxide. And we documented that with looking at using the electron dispersive capability of the scanning electron microscope, and we can see the vanadium peak that's in there. Okay, so here is some of the moon milk with, if you look really closely, there's a little bit of the yellow gold of the vanadium oxides. And here, the organisms, there are two that really dominate One is a hydrogen eater and one is a methane eater. So things really differ in different areas, and that's why looking at different formations and what can give the best signal of life is a really good thing. So one of the things that Jenny Hathaway in my lab did is she compared the microbiology that we were getting in the Azores on the island of Terceira to that we're getting in Hawaii. And what you can see in this lower right in this statistical analysis is that the samples from Hawaii are quite a bit different than the samples from Terceira. So different organisms living in different places. And then if you look up here, we also added the component of how different elements differ. And one of the things is why Hawaii has a lot more copper in the basalt that these microbial colonies are on. So big difference. So we're going to take a brief look at the Azores. I worked with Lourdes on a grant from the Portuguese government for 3 years where we studied all the different microbial organisms that are in different parts of the cave. This is a really cool iron oxide formation that formed in this cave in the Azores. This is Jenny Hathaway, who did the work for her master's. And I saw one of the most unusual things I've ever seen in a cave, and that is these little iron oxide hexagons. And Mike Spilday, who is a cave mineralogist, explained that iron oxides do just form naturally this way. So when we looked at the microorganisms, what the hypothesis is that Mike had is that the microorganisms that we're seeing on the basalt and on the edges of some of these hexagons is actually the microbes are actually eating the iron oxide that we see here. So those were really intriguing. And it was something that was in the middle of a path in the volcanic chimney that is a tourist cave. The other thing is, as I mentioned, I got introduced to ooze in the Azores. Here's some of that butterscotch with actually a microbial mat growing on top of it. It also forms a vermiculation pattern in the Azores that was really cool. And now we're going to turn to Iceland, and I'm really, really grateful to Arne Stephensen, who basically made it possible for me to do some research in Iceland. One of the coolest ones is this volcanic chimney there, which you enter on this little platform that's like the platforms people use to wash windows with. And only ours luckily had nice railings around it, which I appreciated. And the guy who took us down actually was able to swing it over to the wall so I could get a sample of the wall communities there. But what we saw there are some really amazing mineral formations, such as what you see there. And we were given permission to do some sampling there. And so for the people that are on that I know have done some work on the blue-green in Iceland, I hope you got to sample in this cave because it has some really amazing things. This is the name of the cave, which I'm not even going to try to pronounce because I will massacre it. But when you look with the scanning electron microscope, again, this is magnified thousands of times, but you can see lots of little microbes in it. And these formations, which look like they're part of the original basalt formation of the cave, actually have a lot of microbial diversity in them. And these are a sulfide. You can see the iron and sulfur peaks here. So this is an iron sulfide. But when you look inside of it, and if you notice up here in the upper right, there is a covering, sort of a biofilm covering here. And underneath it, there are what to me look like mineral-coated microorganisms that are in there. We actually extracted the DNA from a lot of samples across several caves. And what we're looking at here is a plot that shows us how similar or dissimilar different samples are. The more mineral samples, especially these over here on the right, are very different than the biomat samples, and together they are both very different than what you see in the soils, in the surface soils above the cave. So I'm going to end with a segment where we look at the work we've been doing for a NASA P-STAR grant. Jen Blank is the PI on this, and we were basically looking at this microbe mineral continuum and how we could utilize it to help a rover know when it's going through the cave, how do you know where to tell that rover to sample? And in other words, so I know, because I can walk over to the wall, that this crack that you see here in the wall is really a good place to sample. It has these little polyps. And so that's one of the things we were working on. And I know you're going to laugh because this rover is huge, but this is back from 2018. They have a much smaller rover now. But one of the things we did that was super fun to do is we did what's known as a ground truthing experiment. So they gave me 3 20-meter sections, and then they gave me different periods of time to do an initial run through and identify where I think good life is, go through more carefully and really look at those and identify them. And then the last segment of time was to quickly pick my top 5 choices. And then they had a rover do the same thing and compared what I got with the rover. Haven't heard the results of that yet, but I'm still looking forward to that. And the Mineral-Microbe continuum here is similar, but we did include bare rock, which is, by the way, really hard to find. And it turns out there's microbes on all of the bare rock. The coralloids are really well developed here. The mineral crusts are very extensive. And rather than just polyps, like you might see if you had colon cancer, there are actually also fingers. There are some nice cauliflowers and some really beautiful yellow and white and tan microbial mats. And then my favorite, ooze. So if you look at some of the polyps there, and here are the polyps along here, and you'll see these filamentous microbes that are in there. Again, the scale bar here is 20 microns, so these are on the order of 1 micron. Versus looking at some of the knobs on the wall. And one of the things that was most intriguing to me about the knobs on the wall is this is a silica film that is being deposited on the wall. And in fact, there are large deposits of opal A and opal C. And to me, it looks like the microbes are fighting back. They get covered and they poke back up through. And so there's constantly changing going on in the cave. The microbial mats have a really interesting thing about them, and that is that they have a lot of diversity in the microbial shapes. I want to point out to you these sort of fuzzy filaments, I call them. And if you look over here, there's a blow up of it. So you can see the long hair-like extensions on it. And there are spheroid, little tiny spheroids all along them that we're still trying to figure out, but I see this in caves in other places in the world. There are also one of my favorite, what we call beads on a string. And I hope you guys can see my cursor. It looks like somebody took a strand of pearls that have the pearls sort of separated, and you can see the string between them, or the metal cord. And so those beads on a string are something we also find all over the world. This is some of the ooze from Valentine Cave there. And this is probably a spore structure, a spore sac, and you can see the spores from the fungus inside. But then there's all kinds of other different bacterial or other microbial forms that you see here. So if we look at the difference between this by using genetic sequencing, And this is a lot, luckily for you, it's late afternoon, so your brain's fully on. But I'm going to show you the difference between the bacteria and the archaea when you're looking at genetic sequences of who's present. And this is looking at the genus level. So you are a Homo sapiens, so your genus is Homo. And so this is equivalent to that level of hierarchy. And we're looking across the continuum from bare rock, coralloids, mineral crust, polyps, fingers and knobs, cauliflower, microbial mats, and ooze. And one thing you may notice is the ooze is very different. There's some other colored that indicate other genera that are present. And one of the things you might also notice is there are large swatches of the green and I guess flesh colored and pink and gray. These are minor genera, lots of different things that are only present in small amounts, or lots of the actinobacteria that are only present in small amounts, different kinds. But what I want to point out to you is these two, Nitrospira and WB1P19. This is a unnamed genus that was actually originally, the first time I remember seeing it is in a manuscript by Holmes about Wee Bubby Cave. And this is one of the organisms that was discovered in there. Both of these are prominent members of the nitrogen cycle. And you see them as you look at this red and the green across all of these samples. They're a very important and consistent part of it. Now so these are all bacteria. So there are three major, okay. So can you guys see this okay?
Yeah.
All right. So the amazing thing about the archaea, this group of extremophiles, there's several things that are just amazing. But look at all the red stars I have here. All of these guys are involved in nitrogen cycling. And in fact, many of them actually oxidize ammonia, which supplies all kinds of new mysteries. Where is the ammonia or ammonium coming from in the cave? And we have some hypotheses about that based on work of others. And the other thing is, you'll notice several of them have candidatus. Many of these are just newly identified, and they take different roles, like Woe's archaeales actually is known to reduce nitrate. So it has a completely different role in the nitrogen cycle. So really amazing things going on with the archaea. So for anybody who is studying microbes, I highly recommend you look into those. So what we found so far is that their nitrogen and other key elements are really needed by life, and they could be rare on other planets and moons. And so looking in lava caves to understand different ways of looking for life is important, because on something like Mars, where nitrogen is a very few % of the atmosphere, nitrogen is a key element for life as we know it. And so this could be important. Looking at all these different mineral shapes and textures as potential biosignatures for life is really important for being successful at finding life elsewhere. And we're finding out all kinds of new things that microorganisms can do. And just what we found out about what they can do for humans in the last 20 years is simply amazing. So I invite you to ask questions, and because there's lots to be learned here. And thank you very much for your patience with my wretched network this morning. I would have thought it would be better because nobody's watching Netflix.